Good morning, students. In this class, we are going to discuss about Anatomy of Flowering Plants chapter. From the chapter, we are going to discuss about a subtopic Anatomy of Dicotyledons and Monocotyledons plants characteristics. For a better understanding of tissue organization of roots, stems and leaves, it is convenient to study the transverse sections of the mature zones of these organs. So, in that context, we are going to discuss about dicotyledon's root transverse section. So, if you see the particular diagram, it shows a transverse section of sunflower root. The internal tissue organization is as follows. The outermost layer is epidermis, and many of these epidermal cells protrude in the form of unicellular root hairs. So, students, in this class we are going to discuss about some transverse sections of dicotyledons and monocotyledons organs such as roots, stems, leaves and so on. So first and foremost we are going to discuss about dicotyledons root, example sunflower. So that sunflower internal characterization anatomically as follows when we go through transverse section. So let's see a detailed explanation. So the given structure, it means the structure which you are seeing on the screen shows a transverse section of the sunflower root. The internal tissue organization as follows. Let's see what is that. The outermost layer is epidermis. Many of the epidermal cells protrude in the form of unicellular root hairs. You can see root hairs, the first part which is present on the screen. So a thin layer is epidermis. Please watch the screen, watch the diagram carefully, everybody. The cortex consists of several layers of thin-walled parenchyma cells with intercellular spaces. If you see the diagram, in that diagram the green colored tissue is called as cortex with thin walled parenchyma with intercellular spaces. The innermost layer of the cortex is called endodermis. It comprises a single layer of barrel-shaped cells without any intercellular spaces. If you see in the cortex, the inner layer which is present light pinkish color, it is nothing but endodermis, innermost layer of the cortex. It comprises single layer and barrel shaped cells without any intercellular spaces. The tangential as well as radial walls of the endodermal cells have a deposition of water impermeable waxy material suberin in the form of Casparian stripes. This Casparian stripes concept is most important for competitive examination. Please make a note. I repeat, what is this Casparian stripes once again. So the tangential as well as radial walls of the endodermal cells have a deposition of water impermeable and waxy material suberin in the form of Casparian stripes. Then next to endodermis, students, lies a few layers of thick-walled parenchyma cells referred to the pericycle. It is referred as pericycle. You can see just below the endodermis pericycle layer. Then initiation of largest roots and vascular cambium during the secondary growth takes place in these cells. So this is the main role. So during secondary growth, the pericycle plays a major role. Next, the pith is small and inconspicuous. The parenchyma cells which lie between the xylem and the phloem cells are conjunctive tissue. So you can see conjunctive tissue here, which is present between phloem and xylem. There are usually 2 to 4 xylem and phloem patches. You can count here, see students, 4 patches are there. Later, a cambium ring develops between the xylem and phloem. All tissues on the inner side of the endodermis, such as pericycle, next vascular bundles and pith, constitute the stele. So this is very important. Stele is made up of or comprised of, please make a note students, the tissues on the inner side of the endodermis such as pericycle, vascular bundles and pith constitute the stele. This is most important. Next students, we are going to discuss about one more important concept. It is nothing but monocotyledon's root. So please watch this diagram students, everybody. It is very colorful. Hope you are seeing this diagram. The anatomy of the monocotyledon root is similar to the dicot root in many aspects. Then what plant we can take for example here as a exemplar study? Let's take rice, Oryza sativa, or maize, or wheat. So these all belongs to monocots which comprising fibrous root. So if you see that monocotyledon's root, fibrous root, transverse section, it has some specific anatomical characteristics. Then students, let's see what are those. The anatomy of the monocotyledon root is just similar to the dicot root in many aspects. What are those? Let's see. It has epidermis like dicotyledon's root, cortex, endodermis, pericycle, as usual like monocotyledon's root is having like dicotyledon roots. Vascular bundles and pith also present just like dicotyledon's root anatomical structure only, as compared to the dicot root which have fewer xylem bundles. There are usually more than 6 xylem bundles present in the monocotyledon's root. So here in the dicotyledon's root we are seeing 4 patches, but here we are seeing more than 6 patches. So more than 6 patches phenomenon is called as polyarch. So this is the significant characteristic in monocotyledon's root comparatively dicotyledon root. Have you understood? So see this dicotyledon root, in this 4 patches are present, but here 6 patches are present. The phenomenon of consisting more than 6 patches of xylem bundles is called as polyarch. Pith is large and well developed. This is also one of the significant characteristic. Then monocotyledon's roots do not undergo any secondary growth. Understand? You see this one, this diagram very clearly students. Transverse section of monocotyledon's root, example rice, Oryza sativa, or gia, maize, sweet corn. In this root does not undergo any kind of secondary growth, hence they remains smooth and delicate only. This is because monocotyledon's root can be broken very easily comparatively dicotyledon root. You take one dicotyledon root students like mango or neem, etc., tamarind and etc. Then you take monocotyledon roots like this sweet corn, maize, wheat, etc., which is very easy to break. Monocotyledon's root only, because secondary growth is absent, hence secondary growth is lack, we can break it very easily, and strongness and strength is less and weak. Then students, this is one of the significant characteristic in this monocotyledon's root. First point, xylem bundles are many in number, called polyar condition. Comparatively dicotyledon roots, they are 6 in number. Then pith is large and when developed. So these are the two significant characteristics. Then one more, the last one is monocotyledon roots do not undergo any secondary growth. Understand? So these are the roots of dicotyledons and monocotyledons. Next students, we are going to discuss about anatomy of one more important structure. Then the anatomical structure is as follows. Stem. First and foremost characteristic is stem. Now let us see characteristics of dicotyledon stems like sunflower. The transverse section of a typical young dicotyledon stem shows that the epidermis is the outermost protective layer of the stem. You can see the epidermis which is present in this structure and covered with a thin layer of cuticle. It may bear trichomes and a few stomata. The cells arranged in multiple layers between epidermis and pericycle constitute the cortex. It consists of 3 subzones. You can see on the screen students what are those 3 subzones. The outermost one is hypodermis, consists of a few layers of collenchyma cells just below the epidermis, which provides mechanical strength to the young stem. So next one is epidermis, then cortical layers below the hypodermis. So this is the second zone, cortical layers. Below hypodermis consists of rounded thin-walled parenchyma cells, which conspicuous intercellular spaces. Then innermost layer of the cortex is called as endodermis. So this could be the last zone students. So among the 3 zones in the dicotyledon stem, the first one is hypodermis, followed by cortical layers, and the last one is endodermis. Okay, then the cells of the endodermis are rich in starch gains, and the layer is also referred to as the starch sheath. So the endodermis is rich in the starch gains and the layer is also referred to as the starch sheath. Next, pericycle is present on the innermost of the endodermis and above the phloem in the form of semi-lunar patches of sclerenchyma. Got it everybody? So pericycle is present in the inner side of the endodermis and above the phloem in the form of semi-lunar patches of the sclerenchyma. In between the vascular bundles are there a few layers of radially placed parenchyma cells which constitute medullary rays. A large number of vascular bundles are arranged in a ring. Okay, so this is the significant characteristics of dicotyledon stems. I repeat this one, please make a note students, everybody. A large number of vascular bundles are arranged in a ring. You can see in the first diagram in the ground plane structure, or you can see in clearly in a sector enlarged view also. So the ring arrangement of vascular bundles is a characteristic of dicotyledon stem. Please make a note, this point is important for NEET examination. Each vascular bundle is conjoint, open, and with the endark protoxylem. Then open vascular bundles means between phloem tissue and xylem tissue cambium is present. You can see in the structure yellow colored structure. Phloem is blue in color here in this diagram. Xylem is present just below the phloem. Between this phloem and xylem, the yellowish cambium structure is present as a layer that is nothing but vascular cambium. Consisting of vascular cambium is a characteristic of dicotyledon stems. Hence, it is called as conjoint open endark vascular bundles. A large number of rounded parenchyma cells with large intercellular spaces which occupy the central portion of the stem constitute the pith. So you can see below the vascular bundles a large tissue is present, that is pith. Then what kind of tissue it is? Large in number and round in shape and made up of the parenchyma cells and consisting large intercellular spaces which occupy the central portion of the stem is called pith. Understood, students? So this is about dicotyledon stem, exemplar study of sunflower stem. Then next we are going to discuss about monocotyledon stem, exemplar study rice Varieties sativa. Please watch the screen diagram, students. The monocotyledon stem has a sclerenchymatous hypodermis. You can see in the ground view or in the enlarged section view also. A large number of scattered vascular bundles are present. Please watch the screen, students, in the first structure, ground view, in the ground plane structure. I repeat this specific point for NEET examination. The monocotyledon stem has a sclerenchymatous hypodermis and a large number of scattered vascular bundles are present, each surrounded by a sclerenchymatous vascular bundled sheath. A large conspicuous parenchymatous ground tissue is present. You can see in the structure very clearly. Then one more specific point, students. Vascular bundles are conjoint and closed. So conjoint and closed means, can we expect presence of vascular cambium? No, not at all. See the vascular bundles, students. In this structure they mention very clearly. So between the phloem tissue and xylem tissue, vascular cambium is absent. Vascular cambium presents as a separative tissue layer between phloem and xylem respectively. But in this monocotyledon stem, absence of vascular cambium. Hence these vascular bundles are known as conjoint and closed. Please make a note of this point, students. Next, peripheral vascular bundles are generally smaller than the centrally located ones. Have you seen the first diagram, students? Ground plane structure or ground view. Peripheral side, it means border side. The vascular bundles are small in size, right? Then central vascular bundles, have you seen the central vascular bundles, how they are? They're large in size, right? So this is the significant point of monocotyledon stem. Example, rice, maize, sugarcane, etc. Then one more point here, let's discuss about it. The phloem parenchyma is absent and the water containing cavities are present within the vascular bundles only. Understand? So this point we had a discussion in our regular offline class. Hope you remembered it. So this is repetition concept and the two deleted portion. So this deleted portion, hope you understood. I repeat once again, this last point, the phloem parenchyma is absent and water containing cavities are present within the vascular bundles. So students, in this video we had a discussion regarding the deleted portion for board examination. It is nothing but internal structure, it means anatomical structure of dicotyledons and monocotyledon plants, such as for better understanding of tissues organization of roots, stems and leaves. So in that context we learned firstly about dicotyledon roots followed by monocotyledon roots. With that roots anatomical structure explanation is got over. Then we entered into dicotyledon stem and monocotyledon stem anatomical structure explanation. Then with that stems explanation is over. Then in the next video students we will study about anatomical structure explanation of dicotyledon's leaf and monocotyledon's leaf. So students, hope you understood. In this video explanation regarding roots and stem. In the next video, in the next class, we will study about leaves and anatomical study. Thank you one and all.